Welcome to this week's biology department seminar. Today we're very lucky to have Daphna and Joel visiting us from overseas, all the way to the University of Oklahoma. Sorry, I lost track of my notes here, but Daphna and Joel got her PhD in psychology from Tel Aviv University in 1998, and then had an Alon Fellowship for Young Israeli Scientists. And since soon after that has been a faculty member at Tel Aviv University in both the School of Psychological Sciences and the Sagol School of Neuroscience. And among her many other scientific articles, she published one in Proceedings of the National Academy of Sciences in 2015, which really sort of blew open the whole field of human brain studies, in particular with relation to sex and gender, with evidence that she'll be presenting that our brains, most of us, are mosaics of more masculine and more feminine features, as I'm sure you'll hear about. In addition to her scientific publications, she's published a book for a general audience called Gender Mosaic, Beyond the Myth of the Male and Female Brain, as well as a very recent TEDx talk. But I don't want to take any more time away from her talk, so without further ado, here's Dr. Joel. Please help me welcome.
Thank you. I hope this, it's working, right? Yeah. So really thank you, Ari, for bringing me over. And thank you all for coming, really many people, and I'm really glad to see. And I'm going to tell you what I've learned in the past decade about sex, gender, and the brain beyond the binary. And I want to start my journey here with where I started it over a decade ago, with a study on showing that sex effects on the brain may be opposite under different conditions. And I want to start here because I think when you understand this study, the rest of my talk is quite redundant, although I'll give it anyway. But in this study, the researchers reported sex differences in the brain's response to stress. And specifically, they were looking at the density of dendritic spines. So for those of you less familiar, do you have some kind of a pointer? So for those of you less familiar with the brain, what we see here on the right is a neuron, and you see the extensions, green extensions are the dendrites, and along the dendrites you see small red dots. So these are the dendritic spines, and I'm not going to explain what they are doing. We're just looking at the anatomy, at the structure now of the brain. So they measured how the dendritic spines, or how the density changes in response to stress. And they noticed a sex difference. So whereas in males, the density of spines was increases. So you see now the line there. Maybe this will help me. Okay, so this is the dendrite. This was green in the previous picture. And you see the extensions, the small extensions, these are the dendritic spines. So you can see that in the males, the density was increasing following stress. But in females, the opposite was happening. So for them, this was another example of a sex difference in the brain. But for me, this was something very different. What I saw here, looking this way, not that way, is that sex effects on the brain could be opposite under different conditions. So the sex difference that you see without stress is opposite of the sex difference that you see after 30 minutes of stress. And again, put differently, what's typical of males in the no-stress condition, which is low density of spines, you now see in females under stress. And what we thought was typical of females, that is high density of spines, you now see in the males. In other words, this would be similar to me, for example, going on a way to a lecture, getting stuck in traffic. Happens a lot in Israel. Not sure here it happens a lot, but I'm sure you know the experience. And really, stress that maybe I'll be late for my lecture, but finally getting there on time. This is usually what happens. Even early enough to have time to go to the toilets. going to the toilet, taking my pants off and saying, my penis is back. Now you are laughing because this never happens. So regardless of how stressed I am, my genitalia will not change into the male form, and male genitalia will not change into the female form. But this is analog to what is happening here. So again, running into this study made me rethink everything that I was thinking up until that point about sex and the brain. Another thing that I want you to notice when we look at this slide, and we'll come back to this point later, but I think this is really a very important slide. If you really memorize it, many things, many questions will be answered. We can see here that sex, or something about sex, so I will say sex-related factors, whether this is genes or hormones, we don't know, they need to test it so we can answer, but something about sex, about being female or male, affects the brain. And there are many studies showing that sex-related factors affect brain structure and function. Usually when we hear this, we immediately continue to conclude that this means that there are male brains and female brains. And what I will try to do today is disconnect the two parts. Yes, there is lots of evidence that sex-related factors affect the brain, but no, this doesn't necessarily mean that there are male brains and female brains. And we have done many studies and find out that this is not the case, and I'll show you some of this at least. The other thing that I want you to notice here is that even though sex-related factors clearly affect the brain, knowing the sex category of an individual, that is knowing whether a rat in this case is male or female, doesn't help you to predict brain structure. So knowing that you're going to look at a male rat doesn't help you to predict whether this male rat will have high or low density of spines. To do this, you also need to know the stress history in this example. Okay, so note the difference. The fact that sex-related factors affect the brain doesn't mean that sex category is an important predictor of individual brain structure. And I'll come back to this point later. Okay, so this study and others led me to challenge the sex binary, both regarding thinking of the entire brain and on single brain features, as well as, and I won't have much time to talk about this, but maybe in the Q&A we'll have some time, also to question the binary formulation or thinking of gender and of gender identity and of sex itself. And it's not really easy to challenge the gender binary because, as Veronica Sands really nicely put it, the problem with the sex binary is that there has never been an hypothesis or a theory to test. It is an epistemological framework that runs behind, above, and beyond particular theories and research projects. And if we think about the brain, then scientists use the binary framework or talk about male and female brain, but different scientists mean different things. So some may think of male and female brains as two distinct systems. Others will talk about the typical male brain being different from the typical female brain. Others think of brains as aligned along a continuum between male and female, such as a Baron-Cohen extreme male brain hypothesis of autism. And still others, this is more recent, think about brain structure as male or female because brain structure can be used to predict whether the brain owner is male or female. So we see different formulations, very different formulations of the idea of male and female brain. And so what I want to do is go back to where the binary comes from and where thinking of the binary comes from, and this is the reproductive system or the genitalia, where we do understand what we mean when we talk about male and female genitalia. So what you have here is on the right is a piece of art of an artist, Jamie McCartney, and on the left, this is an equivalent Great Wall of male genitalia, if you want, that I've created using Google Images. So you can imagine the types of ads I get since. But the reason that I have these here is that I want you to appreciate that although there is within sex category variability, so we don't all look the same, the genital organs of most individuals come in one of two distinct sets, Either we are born with only male typical organs or only with female typical organs. And it is quite rare that a baby is born with genitalia that are not clearly male or female, either because one or more organs are in a form intermediate between the male typical and the female typical, or because they have some male typical organs and other female typical organs. So these will be called intersex or DSD, okay, not male or female, the genitalia, not male or female. But it's quite rare in the genitalia. Now with this understanding of what it means to have male or female genitalia, this is brought into, usually implicitly, into thinking about sex effects in other domains. And two important biases affect the way we think about sex in other domains. One is dimorphism. So here what you can see is that each organ has two distinct forms. So it's very easy to differentiate usually between a clitoris and a penis, even though they are both derived from the same origin in the same tissue in the fetus. So this idea that things come in two forms. So this is one bias. And the other, which I'll focus more about, is that sex effects or sex differences add up to create two systems, which is clearly the case in the genitalia. So you have one organ, another organ, and then it comes in two sets. And I want to start with this second bias about how sex effects add or do not add in whole systems. And I want to illustrate the problem, because this is usually an implicit assumption, it's not explicit, with an anonymous reviewer that I think you like. So he starts, or I don't know, I guess it's he, but it's not fair. So they start with the usefulness of the dichotomization between male and female brain stems not only from its biological reality. So, okay, we've done, right? So if it's biological reality, why should we have all this conversation? So this is just an example of what Sand says. The assumption that sex is a binary was never questioned because it was never an hypothesis. It was a taken for granted starting point. So this reviewer's comment really illustrates this. So they start from the fact that this is biological reality, and then they continue. But from the fact that sex differences of all sizes and kinds exist at every level of brain function and anatomy. Okay, so the point, and this is a common mistake, that because there are many differences between the brains of humans, men and women, then there must be two systems. And they continue. The collective evidence, referring to sex differences, still overwhelmingly warrants categorization of brains as belonging to different, if often overlapping, populations, i.e. female and male brains. Okay, so what I want you to take from here is the title in the beginning. So it's very common to assume that sex effects or sex differences on the brain add up to create two systems, just as sex differences in the genitalia most often do. But this is not an explicit assumption, it is an implicit assumption. And I can demonstrate, or it's very well demonstrated in the summary image of this study, which probably some of you know, got a lot of publicity. So in this study, what the researchers did is look at the strengths of over 9,000 connections in the human brain. And they depicted here all the connections that showed a difference between men and women on average. So several dozens. Of course, you can imagine that even though only several dozens of the connections out of 9,000 showed significant differences, this was the title and the entire focus of the paper. But this we'll put on the side. Okay, so what we have here. So in the upper panel, we see all the connections that were stronger on average in men compared to women. On the lower panel, all the connections that were stronger on average in women compared to men. The color code, blue for intra-hemispheric connections and orange for interhemispheric connections. And the text that accompanies the figure talks about male brain being like this and female brain being like that. So the way the researchers describe or interpret the data is that the upper panel represents the typical male brain and the lower panel the typical female brain. So they are using the logic of the genitalia, assuming that what's typical of the brains of males is to have male typical features And what's typical of the brains of females is to have female typical features. Actually, it sounds quite common sensey, right? You know, what else could you expect? But it's not enough to have common sense. So you need to test this, and they never did. They simply assumed that this is the case, as the anonymous reviewer in the previous slide. Now, why suspect that this is not the case? Because of what I showed you before, because sex effects may be opposite under different conditions. And what I'll show you within the next few minutes is that the result of these interactions between sex and other factors, in fact, there are no brains like this. I know because we tested. So there are no blue brains and orange brains like this. What's typical of humans is to have a combination, each a unique combination of connections that are more typical of men and connections that are more typical of women. So each brain has a unique mosaic of these blue and orange connections. Okay, so we'll get there. But I just want to show you when we start to, not to a priori assume the binary, we may end up in interesting places. This type of interaction that I've just shown you between, with the rats, between sex category and other factors have been demonstrated for many regions of the brain, for many features of the brain, and following many different manipulations. I showed you the first slide was acute stress, but it can be many types of environmental manipulation. It could be stress to the dam, the pregnant rat, and then you can see these effects in the offspring when they are old or adults. It can be simply housing the rats, one to a cage compared to several to a cage. This will reverse some sex differences in the cortex. Okay, so many different manipulations. And when you look at all these studies together, as I have done over a decade ago, what you realize is that these different manipulations do not flip the entire brain from the male form to the female form or vice versa. They affect only some features. So even if I assume that at some specific point, let's say now, my entire brain is in the female typical form, following such a manipulation, some of my brain features will change the form, others will not, and I will end up with a mosaic brain. Mosaic in terms of having both female typical and male typical features. If we were talking about genitalia, then we will call it intersex genitalia. Okay? This was a term I was using at the beginning, but I received a request from intersex activists not to use this term, so immediately I stopped using it, and I'm using mosaic instead. But, so I'm just doing the connection so we know what we're talking about. So this was 2011, and it took me a few years to understand how I can test the mosaic hypothesis in the human brain. And together with a colleague from Tel Aviv University, Yaniv Asaf, and a group of students, we analyzed the structure of over 1,400 human brains and used MRI images. And we did different analyses, analyzing gray matter, white matter, connectivity. I'm going to show you only one analysis. And this is a study, PNAS study, Ari mentioned in the beginning. So in this analysis, we divided each brain into 116 regions of gray matter, measured the volume of each region, and wrote it down in a table. In this table, each line represents the 116 volumes of each region. To appreciate similarities and differences between the brains of men and women, we then used a color code in which we looked at each brain region, and if this region was relatively large compared to this region in all other brains, men and women combined, then we painted it green. If it was relatively small, we painted it yellow. Okay, so we painted in this way the entire table, and this is what we got at the end. So on the left, you see brains of men. On the right, brains of women. Each line, a single brain. Now, you don't need statistics. Don't tell the teachers. You don't need statistics to see that there are differences between men and women. At the group level, there is more green at the women's side and more yellow at the men's side. But when you look at each brain, there are no green and yellow brains. Instead, each brain is a unique combination of green, which is more common in women, and yellow, which is more common in men. So again, I want to stress that the question for me is not whether there are sex-gender differences in the human brain. There are, you can see them here. I'm also not interested in the question of how they got there, so the nature versus nurture question, but only on whether these differences add up consistently within each brain to create male or female brains, as is the case in almost always in the genitalia, and as is assumed to be the case also in the human brain, or whether brains are mosaics. And I think that when you look at these images, the answer is quite clear. So I'll go back to these images and to thinking, okay, if brains are not male or female, then how best to describe the relations between sex and the brain? So I'll come back to this question in a few minutes. But before I do, I just want to diverge for a minute and talk about gender. So what the brain does, not just how it is built. So we use a similar approach to ask, are there men and women? So do men and women come in two distinct forms in terms of their psychological characteristics, behaviors, etc.? And we all know that there are group-level differences in many of these measures. So what we did is take open data sets, look for large gender differences in these data sets, like here, what you see here, and then for each of these variables, defined the feminine and the masculine sides of each variable, or scores of each variable. So, for example, if in this sample, if someone had high self-esteem, what will be the color? Pink or blue? High self-esteem? Blue, yes. I mean, you couldn't know what we found in this specific sample, but it's a quite common finding that men, on average, have higher self-esteem compared to women. So high self-esteem would be masculine, would be blue. Low self-esteem would be feminine. If someone was really worried about their weight, color, pink, okay? Because women, on average, worry more about their weight compared to men. So we go like this for every variable, 7 in this example. So what you see here, men on the left, women on the right. Each line here is 7 variables. And again, you can easily see the group level differences. So there is more blue at the men's side and more pink at the women's side. It has to be this way. This is how we defined masculine and feminine, blue and pink. You can also see that there is quite a lot of pink in the blue and quite a lot of blue in the pink. Again, a very common finding. For any psychological variable, there is some overlap between men and women. So some men will have the feminine characteristics and some women will have masculine characteristics. This was all known, not interesting for us. What we asked is whether there are blue and pink humans. And there was not even a single person that had all several characteristics in the feminine or masculine forms. So most humans had a mosaic. They had some variables in the feminine form and others in the masculine form. So we can see that also humans, in terms of the gender characteristics, do not come into distinct types, men and women. And we've recently launched a website in which you can actually see and explore your own gender mosaic. So you answer a set of questions, and then the website gives you your own mosaic. So this is my mosaic. So in each variable, you can see whether you are more feminine or more masculine or neutral. This is yellow. And you can also see how your mosaic changes when your answers are compared to a different sample, because obviously what's masculine and what's feminine differ across cultures. So this is my mosaic when I'm compared to an Israeli sample, and this is to a Japanese sample. So you can see that where I'm feminine and where I'm masculine changes or varies across cultures. So you don't do this now, but you can play with this later. So I want to go back to the brain and ask how best to describe the relations between sex and the brain. And when I look at these images, not only that they don't seem as if they come in two distinct forms, male brains and female brains, it actually seems that we all belong to a single, highly heterogeneous population in which what is typical of all humans is to have both male typical and female typical features, so to have this mosaic. And So I can frame this in a way that will allow me to compare this to the more common hypothesis. So the brain types that are typical of women are also typical of men and vice versa. But there may be some large sex-gender differences in some rare brain types. And you can see this easily if you look at yellow brains. So you can see most people do not have a yellow brain. Most people have a mosaic of yellow and green. But of the people that do have a yellow brain, most of them are men. Okay? So while what's typical of humans is to have this combination of green and yellow, we can see that there may be some differences in rare brain types. Okay? And I want to contrast this with the common view, which I have seen up until now. There are some seats over here, so you don't have to stand in the back. And which is that the brain type typical of males is different from the brain type typical of females. So our first study was to compare these two possibilities, or to ask, are the brain types typical of women the same or different from those typical of men? And our first approach was to use anomaly detection. So anomaly detection algorithms, what you do is you give them a sample of something, let's say a computer activity. This was a first purpose that they were developed. So you give them normal computer activity, and the algorithm learns what your normal activity is on the computer. And then they start running in the background. And whatever the computer does, they ask, is this a normal computer activity or an abnormal computer activity? And if they think it's abnormal, then they will alert you because maybe it's a virus, it's a worm, whatever. Okay, so this is the idea. So we use this, but this time to ask this question. So what we did is give the algorithm or train the algorithm with a set of brains of one sex category, let's say a set of female brains. And then after the algorithm is trained, give the algorithm new brains of males and females, and ask for each brain, is it a normal female brain or is it abnormal? Okay? And of course, then do the same, but train first on male brains. Now, the prediction, if brains are indeed female or male, the way many people believe, then if I train on female brains, then the algorithm will detect many more new female brains as normal than male brains, because there are different populations, right? Okay, this is clear? However, if brains belong to a single highly heterogeneous population, then the algorithm shouldn't really care, because regardless of whether I train on male brains or female brains, I am training on human brains, and the number of males and females that will be categorized as normal should be similar. Okay, so we have very different predictions, almost opposite predictions, which is good in science. So it won't be a quantitative difference. It will be a real qualitative difference in the results. Just to make sure that this approach can detect two populations in biological material when we know that they exist in the data, we use a control that I bought from one of my critiques. So the control is a facial morphology of two primate species. Okay, so there are different species. Let's see if the algorithm can pick it up. In all analysis, we look at the data in the original high dimension space. In this case, it was 190 features, as well as following different methods of dimension reduction, linear and nonlinear. I'm not going to go into this. This is only to make sure that it's something real, or we can generalize about the data set, and it's not specific to how we analyzed it. And I remind you, once we build the model on macaques, and then we build it on capuchins, and I'm going to show you the ratio of each species that were classified as normal out of this species. So let's start with the y-axis. So you can see that all dots fall on the y or x-axis. What does it mean? So let's start with model builds on macaques. So these are the circles. So you can see that the model never thinks that a capuchin, these are capuchins. So the model never thinks that a capuchin is a normal macaque. And when the model is trained on capuchins, it never, so all the points fall on the x-axis, the model never mistakenly thinks that a macaque is a capuchin. Okay, so the model does a very good job in distinguishing between the two primate species. Okay, let's see what happens with the brain. So we do the same, but this time build the model either on brains of males or brains of females. And these are the results. Exact opposite. So all the dots fall across the y equal x axis, not along the y and x axes. Meaning that regardless of whether you train on the brains of males or females, the proportion of males and females that are classified as normal is almost the same. Okay, so it's the exact opposite of what we saw with the primate species. And this is just another data set, so you see that the same idea. So what this shows us is that the brain types that are typical of women are also typical of men and vice versa. And this is why it doesn't matter whether you train on men or women, because in both cases you train of what's typical of humans. What this approach doesn't tell us is why the abnormal brains were marked as abnormal and whether they were marked as abnormal for the same reasons. So it cannot tell us whether rare brain types, rare as opposed to the common brain types, whether they are the same or different in men and women. So to answer this and also to corroborate this analysis, we used another approach, which is unsupervised cluster analysis. And in unsupervised cluster analysis, what you do is give the algorithm all the information about the brains, but you do not give them the algorithm information about whether the brains come from males or females. Okay, so they only have the entire sample. And you ask the algorithm to create clusters. This is where the name comes from. For either two clusters, 3 up to 10. So this is what we did. And then we can, because we know the sex category of the individuals, we can look at these clusters and count how many in each cluster come from men and how many from women. Now again, so we do this with two different algorithms, and again using different methods of dimension reduction. And again, the predictions are very different. So if brains are indeed male or female, then we expect that with two clusters, for example, one would be of mainly brains from women and the other of mainly brains from men. If there are more clusters, then we expect that there would be at least two large clusters, one mainly of men and the other mainly of women. Okay, that's the idea, right? That the typical male brain is different from the typical female brain, so the algorithm should pick it up. However, if brains are all coming from the same heterogeneous population, what we expect is that large clusters, large meaning that they are what's typical of humans, because most humans are found in this large cluster, they will have similar proportions of men and women. However, in small clusters, which correspond to rare brain types, because only few people are there, then we may see large differences. Okay, so again, very different predictions. So what I'm going to show you is the proportions of males or females in each cluster. So the larger of the two, so it will start with from 0.5, okay, as a function of size of cluster. And this is what we get. So with large clusters, which have most of the sample, you can see that the proportion is almost half. So a very similar number of men and women in the large cluster. However, in the small clusters, very small clusters, so less than 100 brains, we can see that many clusters have a similar proportion, but some clusters show very large sex-gender differences. So for example, here, it will be 6 times more from one sex category compared to the other. So again, we see that the brain types that are typical of humans are similarly common in men and women. So it's similar to what we saw with the anomaly detection, but with a different approach. But we do see that there could be some large sex-gender differences in rare brain types. Now, why is this interesting or important? For example, if we consider the extreme male brain hypothesis of autism. So this hypothesis takes the observation that there are more males than females that are diagnosed with autism, and then think about this as an extreme case of the male brain. So males in this theory are aligned along a male-female continuum, with the autistic brain being more towards the end of the male side. So this suggests that typical male brains are a little autistic compared to typical female brains. And if we use this way of thinking for other disorders that show, or conditions that show sex-gender differences, and maybe the typical female brain is a little more depressed than the male typical brain, and you can keep doing this. Here, we don't have this assumption. So the fact that there are more people diagnosed with autism that are males than females doesn't mean anything, doesn't tell us anything about the typical male or female brain, which are the same, even though there is a difference in a rare brain type, because most people do not have autism. Okay, so this is a rare brain condition or brain type. Okay, so this allows us a different way to think about sex-gender differences in psychopathology without assuming anything, or without assuming that the typical or the normal or the brain is different in men and women. Another thing that we could do once we had the clusters is to compute the chances that a male and a female would be in the same cluster, that is, would have the same brain type, and compare it to the chances that 2 men or 2 women would be in the same cluster. And we found that the chances are almost exactly the same. This means that the fact that I have female genitalia doesn't mean that my brain is more likely to be similar to the brain of this lady than to the brain of that gentleman. Whereas, of course, this is not the case if we were to consider our reproductive organs. Okay? But sex category, remember the beginning, sex category provides no information about the structure of the brain or how it will be similar or different from someone else's brain. So going back to these images and how best to describe the relations between sex and the brain, the binary framework allows only 2 options. Either men and women are the same or different. There are no more options. And different meaning that there are 2 populations, partly overlapping or not really important here. But when we look at these images, we see a third possibility. And this is that we are all different. And so no 2 brains are the same. And sex category provides little information on how we are similar or different. So this is important because we can also say that in the genitalia, and remember the image that I showed you in the beginning. So in the genitalia also we are all different, right? No 2 genitalia are the same. But sex category provides a lot of information on how 2 genitalia would be similar or different. In the brain, it doesn't. Okay, so there is a huge difference. Now, I wanted to understand more about how to think of brains. And brains reside in a highly multidimensional space. So if we think of these brains, so each brain here is represented by 116 volumes. So it resides in a 116-dimension space. So it's really difficult, right? We can do up to 3 dimensions. So I wanted to understand how brains are organized in multi-dimension space, and maybe this can help me understand the relations between sex category and brains. So this is what we did. So we used the UK Biobank data, which has, you see, around 24,000 brains, so many brains. And we use the data that they provide. And here we are looking at 169 regions of gray matter, so even more regions. And the first thing we wanted to do is just calculate the distance between each 2 brains. Men and men, men and women, women and women. Just calculate the differences between the brains. So what I'm showing you here, and look with me first on the upper panel, is the distribution of distances between every possible pair of 2 brains. And let's look at this together. So let's start with women. Women in red. And the solid line represents the distribution of distances between every pair of 2 women. Okay? The dotted line represents the distribution of distances of every woman from every man. So this is a man-woman pairs. And you can see that the 2 red distributions are exactly one on top of the other. So the distribution of distances between every 2 women is exactly the same as the distribution of distances between every man and woman. And this is also true for men. So if you now look at the blue lines, the solid line and the dotted blue line are exactly overlapping. So it's another way to understand what it means that sex category provides no information. So the chances that, or the distribution of distances of my brain from each one of your brains is the same regardless of whether I divide you into men and women, okay? And this is also true after you reduce the dimension, because this is a characteristic of high dimension. So this is also true after you use PCA, principal component analysis, to reduce the dimensionality of the data set. Another way to think about this is to look at the distances between every brain and the average male brain and the average female brain. So this is the distribution now not between two brains, but between every brain and the average brain. And one thing that first I want you to concentrate or focus on is where the brains start. So when we look at a single dimension and we look at a normal distribution, most observations are around the mean, right? It looks like this, and most observations are around the mean. You go 2 standard deviation to either side, and most of the observations will be there. Here, the scale below is in standard deviations. So you can see that brains start at around, I don't know, like 8 standard deviation from the mean. So multi-dimension space behave very different from single-dimension space. What we have is you have the mean, the average, and it's empty around it. It's empty for a very long time. You go and you go and you go. 10 standard deviations, then the brain starts. So let's think of this in 3 dimensions. We can also think of it in high dimension. Okay, so it's like the middle of the ball is empty for a long time, and then brain starts. And you can see that the density of the brain increases, and then it decreases again. So we can think of it as like a balloon. Okay, so all the observations are on the periphery of this balloon, and this is what you see here. Now let's go to the sex differences. So let's start again with women in red. So you can see that the distribution of distances between each female brain and the average female brain is a solid line, and the average male brain in dashed line are completely overlapping. So the brains of women are similarly far from the average male brain as they are from the average female brain. And this is also true for the brains of men. And there is a difference between the average male and female brain, and it's marked here. This is the size of the difference. This is around 1 standard deviation, which is quite a large difference in single dimension space. So this would be the extent of overlap if we were looking at a single dimension. Okay? But in a high dimension, as brains are, there is no meaning to this difference in the average. There is no one there, and they provide no information about the distances between or about male and female brains. So I hope this gives you more understanding. At least it helped me to understand what I mean is we are all different. You can see we are so far apart. The differences between brains are in 10, 15, 20 standard deviations. We are very far apart one from another, and sex category gives you no information about how you differ or similar to someone else's brain. Okay, so Ari, you tell me whether to continue or to stop for questions.
5 more minutes.
5 more minutes, okay. Okay, so the last thing I want to show you, it's a little complex, but bear with me, I think it's interesting. Up until now, we were talking about whole brains. Now I want to go back to the level of single brain features, and again show you how the question that the binary framework allows us, are women and men the same or different, that is, belong to a single distribution or to 2 distribution, how it restricts our ability to really understand the relations between sex and the brain. So in a conventional analysis by sex, let's say t-test, which you're all familiar with, the null hypothesis is that men and women come from a single distribution. So let's say that this was in blue, the distribution of some specific measure of the brain, the size of the amygdala, whatever, doesn't matter, okay? And in orange, it will be the distribution of this measure for the size of the amygdala in women. Then a t-test, what it does is ask, does these distributions, observed distribution, come from a single population, which is marked here in the blue line, or from 2 different populations? which are marked here with a solid line and a dashed line. Okay? So men from a male distribution and women from a female distribution. However, if you remember, I always come back to this. If you remember this, then we can together think of a third possibility. And this is that both men and women, or in this case male and female rats, both male and female rats can express the two forms of these dendrites. They can either show high density or low density, but at under different conditions, for example, or in different proportions. So this allows a different way to describe the same observed data, as if they belong to two distributions, solid one and dashed line, but men and women sample with different probabilities from the different distribution, which is marked here with a red and blue. So this is one distribution. This is our female and male sampling from it. And this is the other distribution, males and females sampled from this. So a conventional analysis by sex, such as t-test, does not consider this possibility. So this is not an answer we can receive because we never asked this question. But there are additional statistical methods, and we use mixture analysis, which allows to ask exactly this. So a mixture analysis can compare, or in the mixture analysis we conducted, the null hypothesis is that females and males sample from a female and male distribution. So this is the alternative hypothesis here, is a null hypothesis now. And the alternative hypothesis is that male and female sample from the same two distributions. And we used, again, the data from the UK Biobank, now over 289 MRI-derived measure, to answer this. Because for the mixture analysis, you need large samples. When you do a simple t-test, what you find is that the null hypothesis is rejected in about 90% of the cases. So if we were doing only this, for 32 brain measures, we will conclude that men and women sample from the same distribution. And for the others, we will conclude that they sample from a male and a female distribution. However, when we did the mixture analysis, we found that for 99% of the measures, this description was rejected in favor of the alternative, which is that men and women sample from the same two distributions. And when we looked at the other actual probabilities of sampling from the two distributions, so what you see here is P is a chances or probabilities that a male will sample from the high mean distribution, and Q is a probability that a female will sample from the high mean distribution. And you can see that almost all the dots fall along the y equal x axis, meaning that actually they look like this, not like that. So both men and women favor the same distribution. So either both men and women prefer the high mean distribution or the low mean distribution. So again, if we ask, do men and women belong to two populations or to one, the answer is neither. The best description of brain features is that men and women sample from the same two distribution, not from a male and a female distribution. The last thing I'll show you, and I'll end with this, is that we could also use the few examples of cases in which men favored one distribution and women favored the other distribution. So these are all the dots that fall on these two quarters. And for these, where there is a clear male-favored and a clear female-favored distribution, we can ask, are brains consistent in sampling only from the male-female distribution or only from the female-favored distribution? So we can go back to the mosaic question. So are brains indeed blue or orange, right? Or are they mosaic? Some feature will be sampled from this distribution, other from the other. So what we did here, we looked at the correlation between the posterior probability to sample from the male favored distribution across measures. And you can see the correlations in men on the right triangle and the correlations in women in the lower triangle. And you can see that most correlations are around 0, meaning that the fact that you sample from the male favored distribution of one measure doesn't give you any information of whether the male or female favored distribution were sampled of another measure. Okay, so again, we see the same type of result, but using a very, very different mathematical or analytical method. The last thing I want to point out is that you can see that the two triangles are almost identical. Okay, they look as if they're just a copy one of the other, which was very interesting, wasn't expected specifically. So what we looked at is all correlations between every two pairs of measures. And these are in blue are the correlations in men, sorted according to the size of the correlation. And in red are the correlation in women, sorted as the same as the male are sorted. And you can see that across the brain, on all measures, the correlations between any two measures are extremely similar in men and women. So again, using a very different way to understand what it means, the sex category provides very little information on brain structure. Okay? Or in other words, there are no male and female brains. There are human brains. And they are very, very different one from the other. But sex category does not give us information on how they are different or similar. Okay, so I'll stop here. Thank you. Yes.
Yes. So I was curious as to whether you took into account or looked at gender identity. Were you looking at men and women's brains, looking purely at male-female, or were you looking at the gender identity there?
Yeah. So excellent question. So all these studies are using available data sets in which they are all self-identified. They didn't ask about gender identity, so they actually asked them to mark the gender. So I wouldn't know whether this would be equivalent to gender identity. Okay? But this is how they chose to present themselves. Yes?
I'm wondering, so like very convinced that there's just a human brain, but I'm wondering how do you put this into practice when you're designing experiments or doing analyses now? Like, do you not collect data on sex? You still include it? Like, what should I be doing? Should we have a category for each of those 168 brain volumes?
No.
Like, what should we do?
Okay, so excellent question. Just repeat. So, okay, we understand. So what should we do now? Not collect data on sex category or gender identity, etc. So the answer is, and this is important, first, always use the two sex categories when you collect data. So do not do studies on only one group, only on men or only on women. And probably most of you are familiar with the NIH call to consider sex as a biological variable. So one part of it I think I strongly support, and this is that you always have to include males and females in animals and in humans, regardless of how you define this. But the question is whether to use sex category as an independent measure when you analyze the data. And what our studies clearly show is that if you study normal brain function, for example, so you are interested in the neural subset of language, do not add sex category into your data, because it will only lead to false positive results. Because what you are doing is you are sampling for a huge, very variable population, usually very small samples, comparing them. You'll find differences, but it would not be replicated in other studies, and this is an obvious recurring problem in functional MRI studies, probably because of this. So don't, it will just lead you the wrong way, okay? And what we need to do, and what I managed to do, is show that sex category is not a good predictor of human brain variability, but maybe it's time to start looking for the variables that are important. Socioeconomic status, experiences, education, things that also we can change or, So this may be this important. We can ask many questions of how, do brains belong to different categories? How best to describe them are very important questions, whether different categorizations relate to specific conditions, specific psychopathology. These are all great questions. But in order to ask them, first we need to put aside the belief, very strong belief, that the most important thing about human brains is sex category. And then start studying. And if for some domains we'll find out that sex category is important, great, no problem with this. But turn this into an empirical question rather than an a priori assumption. That you can do the work that I have done, and regarding brain structure and typical individuals, you can just ignore this. But in other domains, this may be important. Okay, so it's an empirical question. Okay, this is, I think, the most important thing. Yes?
So when you look at hundreds or thousands of characteristics of brain area volumes or something, clearly you get this mosaic pattern. But were there any of those characteristics that were individually consistently associated with one sex or another?
Well, not in humans. So, I mean, okay, again, many features you can see differences. So at the level of the single feature, for example, one form will be more prevalent in men and another will be more prevalent in women. But when you look at several together, you always see mosaics. And this is true for MRI data, and we also did a very similar analysis with the data of the human hypothalamus using Dick's Web data on regions that show very, very large differences between men and women, much larger than what the differences that we typically see in MRI. And again, also there, we saw that mosaic is, many brains are mosaics. So also with brain features showing very large differences. And you may want to remember that in the human brain, differences between men and women are usually not very large in comparison to other species, like, for example, rats or mice, when you can see, in some measures, much larger differences. Yes?
A follow-up on this question, if I understood it correctly, could you use sex to, I guess, predict certain areas of volume that, like, if I understood, do differ when you look at those specific spots rather than the whole brain?
So for every single feature that shows a sex-gender difference, the best prediction would be according to your sex category. So let's move from the brain for gender characteristics. It's easier to understand. So for example, if I need to predict whether you love football or not, because I guess in the United States men favor football more than women, I guess that you are a man, then I would predict that you love football. And then with another feature, again, the best prediction for single features is going with your sex-gender category. But I will end up, if I were to predict 10 activities that you prefer, I would predict that all 10 you are masculine in them. Obviously, I would be wrong, because it's very, very rare that someone has 10 masculine characteristics. So at a single level, yes, but when I need to predict several characteristics, I will be wrong. And I have no way, the only thing that I can predict is that most chances that you will have more masculine than feminine characteristics. It's not true for everyone, but on average, this will be true. But it doesn't leave me with much, right? Because 6-4 is more, 9-1 is more, other combinations, okay? And I do not know in what you are masculine and in what you are feminine. And this, of course, is the most important factor, right? Your specific mosaic. So this is a distinction between single variables and going to the whole brain or whole personality, etc. Yes?
Have you done any research on looking at...
Can you, it's really difficult. Yeah, thank you.
Have you looked at juvenile brains or brains over age categories?
Yeah, so with humans, there is a very large data set of neonates in humans, and I was really looking forward to analyze this and created collaboration. But then it turns out that except for the difference in size, which already in neonates you see it in humans, there are no differences. So there was no mosaic, because if there are no differences. So most differences in humans appear in adolescence and later. In our sample, most of these studies are 18 and above, so it's not very informative. We are now doing something in rats, and then they have juvenile. And it seems that mosaicism is similar in the two age groups. But this is something that needs to be studied more carefully. But up until now, I don't have good evidence that it changes throughout life. Yes?
Have you drawn out, or do you have reflections on the implications of your research for the transgender identity?
Can you be more specific?
Well, in other words, does your research suggest an explanation for people who are transgender? And, in other words, the physical is different from their experience, their identity as male or female.
Okay, so very good question. So first, it's a good opportunity to say that what we find in the brain can tell us nothing about causality. So even if we find differences between men and women, gay and straight, trans and cis, whatever categories we compare, we can never say whether this difference is the cause of the behavioral difference or identity difference, or is a result of this. Okay, so the nature-nurture question can never be answered by looking at a brain, because the brain changes. And what we see now is a result of the interaction of nature and nurture. Okay, so this cannot answer the question. What I can say is first that there is no male and female brain. So this is true for cisgender individuals and transgender individuals. And I can say this because we analyzed Dick-Swaib data, had some transgender women, and cisgender men. And all three groups showed mosaicism. So I guess, like everyone else, mosaicism, this is what's typical of humans, all humans, regardless of gender identity. And I can also say that there are a few studies on the brains of transgender individuals, comparing them to same gender, self-identified gender. And again, from the data that we see, and I was not able to analyze it with the mosaic framework, but from the large variabilities that we see, I think that we will also see this in MRI data, that we'll see this mosaicism. And also that the effects of hormones, because hormones obviously affect the brain of everyone. It doesn't matter if it's endogenous hormones or exogenous hormones that affect the brain. We know this in humans and we know this in animals. Then how they affect the brain depends on other factors. So also studies looking at brains of transgender individuals before and after treatment, hormonal treatment, again, they see huge variability in the changes. And again, they haven't looked at internal consistency, but I guess what they find is the same pattern, that because sex affects the brain in ways that interact with other factors, then we have this, it's never consistent. Okay, so I hope this answers. Yes?
So hormones can turn genes on. Anybody looking at the animal model system at gender-specific related behavioral changes transcriptomics?
So I don't know about this, but again, I want to stress, hormones affect the brain. Now, as we see it here, our hormones, whatever is circulated in our blood, affects the brain. But how they affect the brain depends on other factors. So two men, for example, with the same testosterone level, undergoing the similar experience may react differently because of other factors. This is the important thing to remember. Okay? So I don't know about studies that tested this. Very interesting to test, but okay.
All right, well, if there's no more questions right now, let's thank Dr. Joel again.
Thank you.